Hello, and thank you for downloading this presentation on clinical trials for biological medicines. My name is Ciara McDermott. I'm a pharmacist. I have previously worked in industry and I'm currently working in hospital. My job in industry included working as a clinical research associate for a pharmaceutical company for 5 years, and I'm currently working as a clinical trials hospital in haematology and oncology. The main difference between the two jobs is that a clinical research associate is involved in the setup of a trial in the country and at the individual sites as a liaison between the pharmaceutical company and the site. And a clinical trials pharmacist works at the trial site to order, receive, dispense and reconcile medicines supply and to advise patients on how the medicine should be taken. The industry-sponsored studies are typically registration studies looking at the licensing of new medicines, while academic studies are often looking at a broader real-world evidence. So they use trials with already licensed medicines. This may be gathering evidence of the efficacy and safety of medicines in patient populations in whom the medicine has not previously been studied, or investigating combining treatments to improve outcomes. My aim is to give you an overview of the theory and practice behind clinical trials for medicine and provide some detail on how clinical trials for biological medicines differ from those for traditional medicines. So some of the topics that will be covered are: why do we need clinical trials? How are individual clinical trials designed and conducted? What is the clinical development plan for a medicine? How does a clinical trial or development plan for biologicals, including biosimilars, differ from that for traditional chemical molecules? How is the clinical trial data used in the marketing authorization application used by payers and by clinician? And a look at section 5.1 of the SPC, which is where the clinical trial summary data is stored. Just then to finish, we'll talk about biological medicines as treatments for Irish patients and some insights into careers in the pharmaceuticals industry outside of manufacturing. So the reason that we need clinical trials is to assess the pharmacological effects of medicines, which have the potential to both improve a patient illness and to cause side effects. All medicines exert pharmacological effects, which have the potential to both improve a patient's illness and to cause side effects. The aim of doctors and of the medicines that they use to treat patients is first do no harm. Clinical trials are important as they generate evidence around the quality, efficacy and safety and cost effectiveness of medicines at the population level. Regulation of medicines and medical devices is required to protect and improve public health. This evidence allows regulatory authorities to decide whether a medicine should be licensed, allows payers to decide whether it should be reimbursed, doctors to decide whether they will use the medicine to treat individual patients, and patients themselves to make a decision about whether they will take the medicine that has been prescribed. Clinical trials are very important because we can see the improvement in life expectancy that has happened in Ireland, for example, over the last 40 years. Life expectancy has increased by 20 years. This has to do with general improvements in psychosocial living as well as medicines, but it is a significant increase in life expectancy. Trials, like scientific experiments, are designed to test a hypothesis, that is, to answer the question asked by the primary objective, which is generally a statement of comparison about the efficacy or safety of an intervention A compared to intervention B. For example, intervention A is a new treatment, whereas intervention B is a comparator treatment, either an established medicine or a placebo. The number of patients in a clinical trial depends on a number of factors. Placebo-controlled trials require fewer patients than trials which use active comparators, since the magnitude of the effect will be greater with an active comparator. The reason that clinical trials are more complicated than laboratory experiments, such as science experiments as that we all did in school, is largely because of biases introduced because of the biological variability of human subjects, compared to inanimate test materials in the lab, and also because of the difficulty of controlling the external environment in which humans operate, compared to controlled laboratory conditions. In clinical studies which are used to generate data which a pharmaceutical company will submit as part of a marketing authorization application, these biases are reduced by having a control group who are treated in exactly the same way as the new treatment group, apart from the fact that they receive the different intervention. It's also required to rule out the chance that the results seen in the trial population would not be the results seen in the overall worldwide population of patients who have broadly the same demographic and biological characteristics as the trial population. This is done by including a statistical test in the design of the trial. The gold standard for regulatory trials is therefore trials to be randomized and controlled, but in some instances it would be unethical to conduct randomized controlled trials. For example, if you had a trial to find out whether smoking causes cancer, that would not be ethical. And in these cases, an observational trial design will need to be employed. Clinical trials themselves are designed by expert clinicians who advise on the most relevant efficacy and safety outcomes for the particular disease process. Guidelines provided by the European Medicines Agency and other regulatory authorities detail the clinically relevant endpoints that will need to be measured and the assessments that will need to take place to measure those endpoints. The endpoints may be clinically relevant or they may be surrogate endpoints. Surrogate endpoints are those which can be correlated with a clinical effect without the need for long-term studies to establish efficacy. An example would be using measurements of inflammatory markers instead of clinical flare-up for rheumatoid arthritis. Or another example would be measuring changes in LDL or low-density cholesterol instead of longer-term, larger studies designed to measure the rate of myocardial infarction, for example. The assessments that need to be undertaken to measure the endpoints are defined, and then they are described in the protocol and carried out at each study visit. An important part of clinical studies is reducing the effect of bias, and that can be done by a rational choice of efficacy and safety endpoints, having a control arm, either placebo control or comparator control, drawing up defined inclusion and exclusion criteria within a sample so that results can be transferred from the required population, the patient group requiring treatment, randomizing patients to treatment so that the decision about whether they should be in the comparator group or in the new treatment group is unbiased, blinding the treatment so the patients are not aware of which treatment they're receiving, and ensuring that the length of the trial is rational. The effect of chance in clinical studies can be reduced by calculating the sample size required to demonstrate a difference. This is important to consider the required effect size between the active and the comparator groups. The design may be superiority or non-inferiority and may involve hierarchical testing of endpoints. In a superior clinical trial design, the magnitude of the effect in both the sample and when it's extrapolated to the overall population must be such as to provide certainty that the product is superior to the comparator product. Statistical methods of analysis include baseline population analysis to check that the randomization other treatment assignment was fair and that covariates are equally distributed between groups. Analysis of efficacy and safety endpoints, looking at a statistical test which applies the result of the sample studied in the clinical trial to the population of all patients. Any planned subgroup analysis and how to deal with dropouts. The clinical trial protocol is the document which everybody working on the trial follows. It contains a detailed plan for the trial, and it's a joint effort of the clinicians and statisticians. It's also the basis for the methods section of the report and defines the requirements for the study database, the case report forms which are used to collect the patient data, and also the programming for statistical analysis. Real-world evidence is something that's important, and it goes beyond the clinical trials that are used to register the medicine. Regulatory clinical trials have strictly defined inclusion and exclusion criteria, which limit their applicability to the population of patients that a healthcare professional would like to treat. So after a product has been licensed, there's typically a lack of data in paediatric and elderly patients and patients with serious comorbid conditions. Real-world evidence can be generated by different study designs or analysis, including randomized trials, large simple trials, pragmatic trials, and observational studies, which may be prospective and retrospective. They allow a more broad-based representative population and then can provide evidence to doctors which they can be very valuable in treating their patients. Over the last 15 years in particular, real-world evidence trials have been crucial in demonstrating the comparative safety of biosimilars, which are biological generic medicines. Another topic to consider is how are individual clinical trials designed and conducted. The clinical trials regulation is the legal basis for conducting trials in Europe. It repealed the previous advice on clinical trials, which was the directive from 2001, the Clinical Trials Directive. And the clinical trials regulation is taking up a lot of my working life at the moment, because all trials authorised under the Clinical Trial Directive must end or transition to the clinical trial regulation by the 30th of January 2025. Sponsors must submit initial clinical trial applications from the 31st of January 2023 under the clinical trial regulation. Another place where guidance is found for sponsors who are conducting clinical trials is on Udrelex, Volume 10, which are the clinical trial guidelines. And also in Volume 3, which are scientific guidance for the use for medicinal products for human use. In the scientific guidance, the regulatory authorities provide advice on clinical trials. And they're published all of these guidance for different types of therapeutic areas, and it is extremely useful. They are clinical efficacy and safety guidelines, and they're grouped according to the therapeutic area on the Human Regulatory Research and Development section of the European Medicine Agency website. For example, under clinical efficacy and safety, alimentary tract and metabolism, there's a guideline on clinical investigation of medicinal products for the management of Crohn's disease. The agency strongly encourages applicants and marketing authorization holders to follow these guidelines, and deviations from the advice would need to be justified at the time of submission of the marketing authorization clinical trial. So the guidelines are available in Volume 10 of Udrelex, Volume 3 of Udrelex, Volume 10 contains the headings below. So applications and application forms, safety reporting, pharmaceutical data, information on inspections, additional information and legislation. If there were two key points which summarise how clinical trials should run according to good clinical practice, they are patient protection and data integrity. These are the two pillars on which all clinical trials must be based. So the legislation has evolved over time, starting with the Nuremberg Code, which was published after World War II. The Nuremberg trials in August 1947 heard about war crimes committed during World War II, and it led to the adoption of the Nuremberg Code, which outlined the most important ethical aspects for conducting clinical trials. And that included voluntary consent of the human subject, anticipated results should justify the performance of the trials, all unnecessary physical and mental suffering and injury should be avoided, and the trial should only be conducted by scientifically qualified personnel. The Declaration of Helsinki, which was first published in 1964, was a set of ethical principles developed by the World Medical Association. It was revised a number of times. The last time was in October 2013, and it was further developed to the subjects of the right, safety and well-being of trial patients to make sure they're protected and consistent with the principles that have their origin in the Declaration of Helsinki, that the clinical trial data is credible. ICH-GCP followed in 1990, and then the Clinical Trials Directive in Europe was published in 2001. It describes the requirements for the conduct of a clinical trial in the EU. It, as a directive, had to be implemented individually in each member state, and this became a disadvantage which led to the EU Clinical Trial Regulation. In 2005, the Commission published the GCP directive, and that provided more guidance on procedures for applications to conduct a clinical trial and authorization of a clinical trial by national competent authorities, rules for the protection of participants, rules for reporting of adverse events, and rules on the manufacturing, importation and labelling of the investigational medicinal product. The UDRA-CT database of all clinical trials commenced in the community from the 1st of May 2004, and it was really further to the Clinical Trials Directive. The UDRA-CT database was a confidential database where only competent authorities and the EMA and the Commission can see the data. And as part of the clinical trial regulation, UDRA-CT database is replaced by the Clinical Trials Information System. And the major difference is that this is open to everyone. All members of the public can go to the Clinical Trial Information System and view the data. In general, some of the problems with the Clinical Trial Directive were because it was a directive, member states had to transpose it individually, and this was creating a barrier to the conduct of clinical trials in Europe because member states were introducing additional requirements for their countries. This increased the cost of conducting clinical trials in the EU, and the delay for launching clinical trials increased. In fact, the number of clinical trials applied for in the EU decreased by 25% between 2007 and 2011. And this was a problem for pharmaceutical companies as it slowed down research and made it more costly, but also a problem for patients as they were not getting access to new medicines. There's always a gap or unmet need between what medicines can do and what we need them to do, and so it's a priority for all governments to speed up patient access to new and innovative medicines and to make sure that research can take place in a regulated environment. So the clinical trials regulation was really an effort to simplify and harmonise the administrative requirements of clinical trials in order to allow cost-efficient and safe for patients and reproducible for data integrity clinical research. Some of the major changes introduced by the clinical trials regulation are a single submission dossier to be submitted via a single EU portal, a single decision to be made for all European countries, the new publicly accessible EU clinical trials register, which will be set up and run by the EMA, includes a summary understandable to lay persons of what was found in the clinical trial once it's complete. So the regulation, as we've said, was aimed at fostering innovation and research while helping to avoid unnecessary duplication. It refers to the two pillars of GCP, patient protective, including extra protections for vulnerable patients, such as children and prisoners, and people who have diminished capacity to understand, and also the validity of data. As we said, the Clinical Trials Information System has been established, and it's possible to look up ongoing clinical trials in Europe there now. And the procedure for applying for clinical trials has been streamlined. There's joint assessment by all member states, and then each member state assesses things separately, and then ethics committees participate in the national assessment depending on the national law, but within the overall timeline. So the reason that we have, I suppose, regulation of the clinical trials is patient protection and to make sure that the data is of high quality. And some of the things that lead to this are quality systems within clinical trials, including good clinical practice, the scientific and ethical quality standards, quality management systems, documentation, SOPs and contract management, informed consent so that patients are always fully informed of the clinical studies, procedures and what they are expected to do in the clinical study and their rights in case of any adverse events. The investigation of medicinal product must be to GMP standards. Pharmacovigilance, the importance of safety monitoring, importance of everybody being trained who's working on studies, the sort of validation of computer systems and the requirement to record and report deviations and to assess their impact on patient safety and on the data in the clinical study. So before starting a trial, the protocol must be developed. Then the sponsor will choose suitable investigators and investigative sites. They're often in hospitals. And then the online application is submitted for trials which are since 2023, which are approved under the EU Clinical Trials Regulation on the Clinical Trials Information System. And the type of information that the sponsor must upload are the protocol, the investigator brochure, the dossier of the investigational medicinal product, including certificates of analysis, labelling, TSE and viral safety statements and GMP statements, and a list of ongoing studies with the investigational medicinal product. Then the investigator site staff must be trained. During the trial, the procedures that are ongoing include recruitment, so making sure that the patients who are eligible to enrol in the trial are fully consented and are made aware of all of the facets of the trial before they do that. Randomization, as we mentioned, is needed to avoid bias. Once the patients are enrolled in the study, they will come in at periodic intervals and will be going through the medical checks or investigations. So efficacy and safety measurements will be recorded, and any adverse events will be reported centrally so that they can be assessed on an ongoing basis. The clinical trial staff will also transmit the data to the sponsor for entry into the study database. And there's importantly drug accountability to make sure that there's some evidence that the patients have taken the medicine, if it's oral medicine that they bring home, source data verification, and queries to clean the data. So source data verification refers to the fact that everything that happens in the clinical study is recorded in the patient's hospital record, in addition to being recorded on the study database. And this is really to make sure that there's a complete record in the patient notes of what happened during the study. And it's important for the monitor or the CRA to check that these two records match. So for quality reasons, audits may be run by the sponsor themselves, and also inspections from the regulatory authorities to make sure that the clinical trial quality is there. At the end of the trial, the source data verification and queries must all be finalised to make sure that the data is clean and the database is locked. It's important that clinical trial records are archived safely at the investigator site. The clinical trial regulation requires that all of the records are kept for at least 25 years. Then the statistical review of the trial will begin on the database locked data, and there will be a definition of the different populations in the study. So the safety population is all patients that received one dose of the medication, even if they didn't continue with the full efficacy data that was collected. And then there's various other efficacy populations that are looked at. It's important to then inform authorities and ethics committees that the trial is complete and submit the results for publication within 1 year. So this is where it's possible to view these results on the Clinical Trials Information System registry. One of the reasons that transparency is so important around clinical trial results is because in the past, companies and sponsors did not publish the results of negative clinical trials. And this would mean that the information about whether certain products could be used for different populations or for different indications was not in the public domain. So trials were published always in peer-reviewed medical journals, but there's a publication bias because editors of medical journals may not be so interested in negative trials. So rather than relying on trials to be published in medical journals, we now have the Clinical Trial Information System, which will be a source of publication of trials and their results. And this also helps with the ethical requirement of not performing duplicate trials. This is really important because every clinical trial has a potential risk for the patients that are entering it. So it's unethical to perform the same trials over and over again. So if you wanted to look at clinical trials that are taking place in a particular therapy area or with a particular medication, you can go on to clinicaltrialsregister.eu and also clinicaltrials.gov, which is a US database. However, the above EU website refers to the older registry established under the EU directive. The CTIS website established under the regulation can be accessed by entering https euclinicaltrials.eu. The clinical development plan. Legislation describes what information must be provided by registration clinical trials, and the legal basis for the information which must be provided to obtain a marketing authorization application are from the directives 2001/83 EC and 2003/63 EC. This is an example of the marketing authorization dossier, and you can see that it's separated into a number of different modules. Module 1 is a summary data. Module 2 is the summaries Module 1 is the summary of product characteristics, which is a very short summary of the characteristics or the drug license. Module 2 is the summaries of the other modules, and Module 3 is the quality information. Module 4 is the non-clinical study reports, and Module 5 is the clinical studies. All of the clinical trial results in Module 5 are presented in a systematic way, a bit like the structure of the marketing authorization itself, with a listing and key summary data of all the clinical trials, followed by the details of the trial organised according to the phase of the trial. So the phase 1 sections include the biopharmaceutic, that is the physical and chemical properties of drugs and their dosage, and studies using human biomaterials, pharmacokinetic studies which investigate the effective plasma level of the medicine, and pharmacodynamic studies which investigate the desired biological response. The other parts of the Module 5 include Phase 2, Phase 3 and Phase 4 studies. And then there are various listings. So the clinical trials in human medicines, this is the homepage where all of the guidance can be found about all aspects of study design and also about how to register studies under the Clinical Trials Information System. For the Clinical Development Plan, which is the roadmap which details how the company will design the studies to obtain all of the information that's required, the company will generally start by looking at the unmet need in the therapeutic area and creating a broad wish list, that is the target product profile for what an ideal medicine in this therapeutic area would achieve. The clinical development plan is designed to test how close the actual characteristics of the medicine are to the TPP. It defines endpoints of the various trials, decision points, estimated timelines and budgets. A team of experts from all parts of a pharmaceutical company or an academic sponsor will contribute to the drug development process. Discovery of the drug is the first step, and from there are a series of key decision milestones about whether the product development should continue or not. This includes the discovery project approval, in the non-clinical phase product lead profile approval. A very important milestone is the first human dose, which precedes exploratory clinical, and then regulatory clinical is the start of the phase 1, phase 2 trials up until clinical proof of concept, which is the stop-go decision between phase 2 and phase 3 studies. Once the marketing authorization application is submitted to the regulatory authorities, then there are further clinical studies, for example, the real-world evidence studies that we mentioned, to investigate potentially safety outcomes in a broad population of patients. And once a product is launched, lifecycle management continues, and various requirements of clinical trials may include new indications, etc. Clinical development is a significant part of the cost and a significant part of the time that it takes to bring a medicine to the market. The total cost of developing a new medicine to market is estimated to be in the order of £1.15 billion, British pounds, and takes about 12.5 years. So we can see for the phases 1 to 4 of clinical trials the typical number of participants that are needed and the length of the study. The purpose is also shown on the slide. So phase 1 studies are used to find out the route of administration, safety and dosage. Phase 2 studies are used to find dosing, safety and efficacy and to establish proof of concept. and phase 3 studies look at safety and efficacy with a view of generating the data that is used in the marketing authorization application. In phase 4, the safety and efficacy in real-world populations are examined, and the endpoints related to cost effectiveness may also be included at this phase. As you can see, the arrows show the attrition rate. A huge number of molecules enter discovery, and most are not progressed to clinical development, but there are strict assessments at the end of each phase before the molecule is progressed to the next phase. And we can see that most molecules are lost between phase 1 and phase 2, and then there's slightly less attrition between phase 2 and phase 3, and phase 3 and phase 4. Typically, percentage of losses shown may be due to lack of ability to demonstrate efficacy or lack of ability to demonstrate safety. Every trial has a safety committee from the company scrutinising all adverse events, in particular serious unexpected adverse events that are suspected to be related to the medicine, called SUSARs. There's a requirement to report SUSARs to the regulatory authorities within 7 days of their occurrence. And companies have a safety committee, and authorities also consult about potential closure of clinical trials. I have experienced this and the importance of follow-up of all patients until adverse events are resolved when clinical trials are closed. So looking at the key data to establish during phase 1 studies. They established the most appropriate route of administration for the drug. Most small molecule chemical drugs are given orally, and most biological medicines have to be given by injection or by another route so that the protein components are not denatured by acid in the stomach. PK or pharmacokinetic studies investigate the time course of the drug in the body. The acronym ADME, absorption, distribution, metabolism and excretion, is used, and those processes for the particular drug must be characterized. Bioavailability is also an issue, and pharmacodynamics looks at concentration effect relationship. Bioavailability refers to the extent and rate at which the drug enters the blood. Going back to the first do no harm, the positive biological responses or efficacy must outweigh the negative biological responses, adverse events. The purpose of phase 2 studies is to establish the dose range of the medicine in humans. The starting dose used in phase 1 studies for small molecules is based on the no adverse effect level from animal studies, and as most medicines show both increasing effectiveness and increasing toxicity as the dose increases, these studies are used to define the best dose range. Phase 2 studies also investigate the frequency of dosing. The theory is that the frequency of dosing should be based on the half-life of the medicine, which is a pharmacokinetic measure of how long the medicine is in the blood. But as many medicines with relatively short half-lives can set off a chain of events that lasts for an extended period, example, irreversible blockade of receptors, half-life is not always the key determinant, and issues like convenience, such as once-daily dosing, may often be important. Large phase 3 studies, so-called regulatory clinical studies, are how the company convinced the regulatory authorities of the efficacy of the medicine of the chosen indication. They involve the recruitment of hundreds or thousands of patients. As we have discussed, the efficacy requirement is comparative to either a placebo or a comparator medicine. The gold standard comparative efficacy study is a superiority study. This is necessary for licensing and for payer approval for most new molecules, in particular if the new medicine is expected to be more expensive than the comparator. In a superiority study, the trial is designed to demonstrate that the magnitude of the difference between the groups is large enough, both in the trial, which is the sample population, and after the statistical test is used to apply the sample results to the whole population to say that the new treatment is superior to the comparator. There are some differences in the clinical development plan for biologicals, including biosimilar medicines. Biological medicines are any medicines with a biological origin, whether these are extracted from animal or human sources or manufactured by recombinant DNA technology. Compared to small chemical molecules, biologicals have a much more complex structure and a much more difficult manufacturing process. As there are many different classes of biological medicines, such as those shown on the slide, there is no one-size-fits-all rule for both the data which is required to authorise a medicine and how clinical trials to gather this data should be designed. The main directives are shown on the slide. Advanced therapies such as gene therapy, cell therapy and tissue therapies, including products which are composed of both medicines and medicinal devices, are the most complex. An example is the new CAR T cell therapies for the treatment of certain blood cancers. T cells are taken from a patient's blood, and then the gene for a special receptor called chimeric antigen receptor, or CAR, that binds to certain proteins on the patient's cancer cells are added to the T cells in the laboratory. A large number of the CAR T cells are grown in the laboratory and then given to the patient by infusion. The CAR T cells then attack the patient's cancer cells. This therapy is in current use in Ireland to treat certain blood cancers and is being studied in the treatment of other types of cancers. So the first biologicals were vaccines developed from the work of Edward Jenner in the 1700s. In 1921, the discovery of insulin led to the replacement proteins, and this was followed by hormones like growth hormone. The big problem with these animal and human-derived biologicals was the high risk of infectious pathogens, and methods were developed to remove pathogens from animal-derived products. However, the development of recombinant DNA technology in the 1980s was a huge advance as it allowed safer and more reproducible technology for production. And this included innovative therapies. The new technology made it possible to manufacture proteins which have been difficult to extract, such as epoetin for the stimulation of red blood cell growth and the anti-TNF alpha drugs. And this eventually led to the development of monoclonal antibodies. Antibodies which are tailored to target an array of receptors and pathogens. The monoclonal antibodies have revolutionized treatment of many diseases, most significantly in oncology, example bevacizumab for a range of cancers, and infliximab, an anti-TNF alpha drug used in rheumatology. These medicines are highly specific and have an easily measurable pharmacodynamic action at the drug receptor. Synthesizing mAbs often involves attaching a drug molecule to an antibody. In some cases, the drug molecule was developed as a small molecule, a chemical entity, but did not demonstrate adequate safety to allow it to be used on the market. For example, the anti-CGRP small molecules to treat migraine failed clinical trials due to hepatotoxicity, but when the same molecules were attached to the monoclonal antibody, this allowed the therapeutic effect without the toxicity. In the early days, it was hoped that the specificity of action would result in less adverse effects than in small molecules, and to an extent this is true, but these medicines still have adverse events and safety is still an important consideration in their development. Over non-recombinant biologicals, the main advantages of the recombinant technology are the ability to have large-scale production and reduction in the contamination through infectious pathogens, such as CJD, which led to the withdrawal of derived growth hormone products in the 1980s. This slide summarises the main difference between small molecules and monoclonal antibodies. Small molecules are well defined in structure, whereas MABs are large heterogeneous protein structures. Small molecules can be characterised completely with relatively few analytics, whereas MABs require full characterisation, and it's impossible actually to do full characterisation, but there are extensive analytical packages. And small molecules are produced by synthesis, where MABs are produced in cells, and the process defines the product. I've mentioned about the scientific guidance for clinical trials, which is in volume 3 of UDRLEX. And I suppose in the area of advanced therapy medicinal products, the science and the regulators are moving forward hand in hand. So in addition to publishing this type of guidance, the European Medicines Agency or national competent authorities will meet with companies or sponsored who are hoping to bring a new product to the market to discuss the requirements. And this is a very good mechanism for sponsors. Guidance is not legally binding on either party, but it would be difficult for a sponsor to justify deviating from the advice that was given by a regulatory authority at a later stage. So, some considerations for clinical trials with biologicals. The aim of the clinical development programme is still to establish therapeutic efficacy and safety, and biologicals as proteins are administered parenterally. Phase 1 human safety studies are critical. As biologicals have highly specific effects, their therapeutic potential can be determined relatively easily using surrogate endpoints, and data can be generated in phase 1 that can allow phase 2 and phase 3 studies to be abridged. Considerations in particular relate to complex structure and heterogeneity of the products, biological nature of the products, which can invoke immune responses, and MABs with certain Fc structures may have immunosuppressive properties. Specifically for phase 1 studies, the type of model or animal in which the preclinical studies are done were important. In the development of small molecules, typically there's 3 animal species tested before the molecule moves to human testing, one of whom is not a rodent. And these are important in determining which dose to be used for first-in-man treatment. Using non-clinical studies in this way is more complex for biological molecules, as these complex molecules often have a species-specific action, meaning that the protein is not pharmacologically active in the animal species in which the non-clinical study took place. This led to tragic consequences during a study in Northwick Park in 2006, when 2 patients experienced a CRS, cytokine release syndrome, or a cytokine storm reaction, resulting in the requirements for some of these healthy volunteers to have their fingers and toes amputated. An investigation after this event led to many recommendations to improve the safety of phase 1 clinical studies of biological medicines. The recommendations were published in the Duff Report. One of the major changes was that clinical studies of biological models, instead of using the no adverse effect level in animals as the first human dose, should use a different, more sophisticated measure called the Mabel measure. Just going back to phase 1 studies again, on the other hand, the highly selective molecular specificity of biological molecules means that it can be relatively easy to determine their therapeutic potential using surrogate endpoints. So phase 1 pharmacodynamic studies can be hugely valuable in generating information, and this can reduce the amount of data that needs to be generated from phase 2 studies. An example of this was the clinical trials for the recombinant human insulin. As insulin has a highly specific and easily measurable action, the lowering of blood glucose, and there was a lot of experience with human-derived insulin, evidence from phase 1 studies which showed the recombinant human insulin was very similar to the derived human insulin, provided assurances that reduced the amount of data that had to be collected in phase 2 studies, although some phase 2 and confirmatory phase 3 data were still required for registration. For phase 2 studies then, the generation of a comparatively large amount of data for biological medicines in phase 1 compared to small chemical molecules means that phase 2 can be more targeted and sometimes completely bypassed if enough information was generated in phase 1 to establish the optimum dose and frequency of dosing. The optimum dose level may not be as obvious as with small molecules, because biologicals such as monoclonal antibodies are relatively non-toxic, so it may be difficult to establish a maximum tolerated dose. Sometimes the level at which the efficacy or dose effect levels off is chosen, and sometimes the dose is limited by production capacity. For phase 3 studies, even with the phase 1 human pharmacology studies, confirmatory phase 3 studies are still required to demonstrate efficacy and safety. Placebo-controlled studies are more difficult to justify as medicines are frequently used for the treatment of severe and life-threatening diseases, so an active comparator should be used. And again, blinding may be difficult to maintain due to the highly specific actions of biologicals. In general, biological medicines which demonstrate a dramatic efficacy need smaller phase 3 development program because a difference to comparator treatment can be easily demonstrated. In some cases, the effect is so dramatic that the comparison to historical data can suffice without the need for a comparator. Immunogenicity is a key safety consideration, and antibodies may be binding or neutralizing. However, as for biologicals, the process defines the product. Although phase 3 studies can involve less patients, making them easier than phase 3 studies with small molecule chemical medicines, there can be a problem with the longevity and continuing applicability of the data generated in the phase 3 program. The issue is that the process defines the product, and it's often necessary to change the process, for example, scale up production, changes designed to extend the shelf life of the product, or to change or amend the formulation in some way. And these changes can potentially introduce undetectable alterations in the protein which could impact the safety and efficacy of the product. This means that the value of existing studies may be limited, as there's a need to demonstrate that any change made does not alter the efficacy and safety profile of the medicine, and this needs to be supported by comparability data before the new batches are released. One occasion on which a formulation change had a dramatic effect on safety was when a change was made to the epoetin product Iprex. A change made to the formulation to replace human serum albumin with polysorbate 80 led to an increase in neutralizing antibodies. These antibodies cross-reacted with the endogenous erythropoietin, causing pure red cell aplasia. This was a very rare event, with one patient in 10,000 having this reaction, but it was very serious, and the result was severe anemia, and the only treatment at the time was regular blood transfusions. So as you can see, the patients who had this adverse effect had a life-changing impact. This was a huge shock to the nephrologists that were treating patients with this medicine, and it did cause a large, I suppose, amount of questioning about the safety of biological medicines, and it really kind of led to the requirement for comparability studies when changes are made in the manufacturing process. So the EMA guideline, which talks about comparability of biological projects after manufacturing changes, is an important document. As previously discussed, chemical medicines have a comparatively simple chemical structures and therefore have great stability in the manufacturing process and good consistency between batches manufactured by the same company, and even in the active substances, batches manufactured by different companies, which allows generics to be produced relatively simply. For biologicals, there's much more variability, in particular in the protein folding and the post-translational modifications. Manufacturing conditions such as temperature and pH may also affect the manufacturing process, so there will still be variability in the molecules within a batch and between batches. Any variability identified must be within predefined specifications as defined by the European Medicines Agency. Comparability studies are used to confirm that any differences identified between batches do not affect the safety or the efficacy of the final product. The guideline advises how companies should collect data to demonstrate that changes in the manufacturing process do not adversely affect the quality, efficacy and safety. The extent of the comparability studies required by the EMA depends on the type of manufacturing change applied. Only limited analytical studies may be required following a minor change, while clinical studies may be required following a major change. If clinical studies are required following a major change, it is usually not necessary to repeat the same studies in all indications. The clinical study is normally conducted in one indication and extrapolated to the medicine's other indications. So looking at biologicals, specifically the type of medicines which are biosimilars, and how the clinical development plan for those medicines is generated. A biosimilar is a biological medicine which is highly similar to another biological medicine already marketed in the EU. The concept is the same as that of a generic medicine for simple chemical medicines, although the complex structure of biologicals means that there are more practical difficulties with obtaining regulatory approval for a biosimilar relative to a generic. The advantage of both biosimilars and generics is that they will be introduced to the market at a lower cost, allowing more patients to be treated. This is partly because the manufacturing company does not have to start from scratch with the development and can refer to the existing license of the product already marketed to which it is similar, the so-called reference products. More studies are required for regulatory approval of a biosimilar compared to a generic. It's estimated that it can take up to 8 years and cost over €100 million to develop a biosimilar, compared to 2 years and about €1 to €2 million to develop a generic small molecule chemical medicine. So when can a company designate a medicine as a generic? This is the regulation Directive 2001/83/EC, and a company can introduce a generic if the product is essentially similar to a medicinal product which has been authorised in the community for not less than 6 years. The medicine is marketed in the member state for which the application is made. The applicant should justify the claim of essential similarity, and for generics, an abridged dossier will suffice for the marketing authorisation application. Module 1, Administrative Information, Module 2, Summaries, and Module 3, Quality must be provided, together with bioavailability and bioequivalence data, the first part of Module 5. A company can designate a medicine as a biosimilar if the product is essentially similar to a medicinal product which has been authorised within the community for not less than 10 years, if it's a high technology product. The medicine is marketed in the member state for which the application is made. The applicant can justify the claim of essential similarity. And for small molecule generics, which are the chemical things, medicines, they're relatively easy to copy. There's no formal comparability exercise required. But for biosimilars, which are more difficult to copy, this exercise is required. The same scientific principles that have been in use over the last number of decades to confirm that the similarity of a biological medicine before and after a change in the manufacturing process are used in the purpose of demonstrating the similarity of a biosimilar to the reference medicine. This involves comparability studies and extrapolation of indications. If you look at the difference in approval of a biological medicine versus a biosimilar, you can see that you need, for the original medicine, clinical studies, clinical pharmacology, preclinical studies, and analytical. But there's a paradigm shift in the regulatory approval pathway for biosimilar medicines when compared to the conventional pathway for new biological medicines containing novel active substances. For a novel active substance, the safety and efficacy profile is unknown and must be established before approval. Therefore, the greater emphasis is on pivotal or phase 3 clinical studies to establish clinical effects. For a biosimilar medicine, the focus is on confirming a high level of similarity to another EMA-approved biological medicine containing the same active substance, that is, the reference medicine. The safety and efficacy profile of the active substance in the reference medicine is already established at the time of a biosimilar medicine's development. So clinical trials for biosimilars depend on whether the quality analysis can establish essential similarity. Biosimilars are not equivalent to the originator product. The molecular size, three-dimensional shape, and process impurities differ. So, the comparability quality dossier is required. Because a biosimilar and its reference medicine are made by different manufacturers, additional data from non-clinical and clinical studies are required to confirm there's no significant difference in safety or efficacy. The amount of additional data required to confirm similarity depends on the findings of the preliminary analytical studies. These analytical studies, using state-of-the-art technologies, can measure differences at the molecular level, and they're generally much more sensitive and specific for confirming biosimilarity than clinical studies in human trials. As for any medicinal product, clinical studies of biosimilars are subject to ethics review and scientific review, and unnecessarily duplication of clinical trials should be avoided. When the quality dossier is established, then the manufacturer of the biosimilar will perform a quality comparability with the reference product. And depending on the differences and looking at the safety and efficacy data that's already established for the active substance, the reference medicine, the amount of repetition and new data required can be established. Non-clinical studies, pharmacokinetic and pharmacodynamic studies in animals, repeat toxicity and local tolerance, and clinical studies, pharmacokinetic and pharmacodynamic, typically including one phase 3 equivalent study. The phase 3 clinical studies for biosimilar development are only required to confirm biosimilarity and clinical performance, not to establish the safety and efficacy of the medicine, which is already available from the reference product. This is a schematic which shows the type of information that is needed for the reference medicine and the biosimilar medicine. So for the reference medicine or the original biologic, the purpose of the clinical development plan is to establish the benefit and risk ratio to make sure that the efficacy outweighs the safety. And the purpose of the clinical development plan for biosimilars is to establish essential similarity to a reference product with a known benefit and risk ratio. Some guidelines follow now. European Medicines Agency overview of biosimilars. There's the overarching biosimilar guidelines. And there's also product-specific biosimilar guidelines on the EMA website. From a practical point of view, you can look at the clinical trial information system to find information about clinical trials, or you can also look at section 5.1 of any medicine license or summary of characteristics to see this type of information. So for example, you can see the summary of characteristics for different medicines on medicines.ie, which is the website of the Irish Pharmaceutical Healthcare Association, or on the HPRA website. The HPRA are the competent authority for regulation of medicines in Ireland. The structure of the SPC is shown on the slide, and from our point of view, the most interesting parts are section 4, which is the clinical particulars, and section 5 goes into detail about the clinical studies which are used to generate those clinical particulars. Centralised medicinal products licensed in all of Europe can also be found on the European Union Register of Medical Products. Biological medicines as treatments for Irish patients. If you look at any list of the top 10 molecules supplied in Ireland, for example from the high-tech scheme in 2019, which was published as part of the government spending review in 2021, you can see that there is a large number of biological molecules listed. And the percentage of the budget is shown on this slide, along with the indication. It is a big question about how Europe and Ireland can continue to pay for continued innovation. Biologics are much more expensive than small molecules, and this is why there's so much focus on biosimilars, because it provides a way of providing biological molecules to patients without having to continually pay the higher premium for the innovative product. This data may be slightly out of date, but it says on the slide that 86 biosimilars have been licensed in Europe since 2006, ranging from recombinant proteins to monoclonal antibodies. The Health Products Regulatory Agency, after the Health Pricing and Medicinal Supply of Medicinal Goods Act in 2013, the HPRA have a list of medicines which can be substituted by pharmacists, and they provide the list of interchangeable medicines where pharmacist-led generic substitution is permitted. Biological medicines, including biosimilars, are not included on the current list, so this means that pharmacists are not permitted to substitute one brand of biological medicine for another. Only a prescriber can exchange a brand of biological medicine. However, the European Medicines Agency are keen to move forward the replacements of biosimilars. In the context of biological medicines, the EMA defines interchangeability as a possibility of exchanging one medicine for another medicine that is expected to have the same clinical effect. In clinical practice, this can apply to replacing a reference medicine with biosimilar, or vice versa, or replacing one biosimilar with another. Switching is when replacement is prescriber-led, and substitution is when the replacement occurs at the pharmacy level without the prescriber being contacted. So at the moment, most of the biosimilar is prescribing, and generic medicines may be switched by community pharmacists. So just to talk a little bit about the different roles that are available in the pharmaceutical industry in Ireland. There are roles in commercial, in sales and marketing. Overall, we know that 45,000 people are employed in the pharmaceutical industry in Ireland, and the majority, of course, are employed in GMP environment, such as the manufacture of API and finished pharmaceutical product for export. But there are also some jobs in the domestic sales and marketing part of pharmaceutical companies. And really, these roles are designed to make sure that doctors are informed about medicines that are available. The aim of these staff, commercial, in sales and marketing, is to inform doctors about the right medicine for the right patient at the right time. There's strict rules about how promotion is carried out, and activities and materials must all be signed out by a regulatory compliance part of pharmaceutical companies. The type of jobs available are sales rep, marketing person, MSL or medical science liaison, which typically works at the stage of a medicine coming onto the market. And there are also other roles as well. Non-commercial roles in pharmaceutical companies include those in good distribution practice, or GDP. There's roles available in clinical trials, from regulatory affairs in companies, registering clinical trials, pharmacovigilance in safety, medicines information, medicine scientific liaison, which typically is somebody who discusses new products with physicians, finding out how the physicians treat their patients, and also asking doctors about whether the profile of the medicine being developed by the company would be a fit for their patients. Medical signatory people are the persons who sign off the compliance of the activities and the materials for the entire company. Pharmacoeconomics is an important area where medicines are being presented to payers. Pharmacoeconomic data looking at outcomes of treatment may be used to justify more expensive medicines if a company can demonstrate that there were savings made, for example, in hospital admissions, etc. And then, of course, there's supply chain and finance as well. So thank you very much for listening to this presentation. If you have any questions, I can be contacted on my Gmail address on slide 1. Thank you.